medicine has to prevent as well as cure or treat. And in fact, possibly, it is easier and less expensive to prevent than to cure. And this is the case of many of the new diseases which are linked both to epidemics but also to chronic diseases. And this is linked to the fact that our world is becoming more globalized, that there is a uncontrolled demographic growth in many countries with a concentration in large cities. Likewise, there are problems which are linked to nutrition, the fact that we use a lot of pesticides, and that very often there is a water shortage, as well as the fact that there are some new environmental factors that have to be taken on board, in particular the problem of greenhouse effect and a warmer climate, and also contamination by electromagnetic radiation, which we use all the time, even now here today. Then there is the problem that wild animals have less and less land, and therefore they are more and more in contact with farm animals and with ourselves. And this also means that there is a transfer of viruses from wild animals to domestic or farm animals and then on to human beings. Because of all these reasons, our immune systems, which are our main defense against viruses, bacteria, parasites, has a lot of work to do. And we have inherited from our ancestors good defenses. We are the survivors. We've been deselected from cholera. We're the ones who've survived the plague. But because of all the aforementioned phenomena, our immune systems sometimes are experiencing decline. And we live for longer and longer. We've learned about 30%. We've gained 30% as compared to the previous century in terms of life expectancy. And life expectancy, as it is now, means that there are very many older people who are more at risk for infectious and parasite diseases because of the decline of their immune systems, which is age-related. I would also like to stress the following points, which are chronic diseases that I have listed: cancer, diabetes, cardiovascular diseases, neurodegenerative diseases, and chronic fatigue syndrome. What do all these diseases share? Well, they're multifactorial to start with, and each one has a specific use, but they also share two things: a biochemical symptom and an infectious one.
Donc, il y a deux facteurs communs.
So there are two factors, a biochemical one, for example, oxidative stress, and infectious agents that might be latent. This is summarized here in that all the factors of these diseases are summed up, and they create oxidative stress. Sometimes there are infections, bacterial and viral infections that may develop. This is also summarized in this slide, in that there is an unbalance between our antioxidant activities and types of molecules that are oxygen derivatives and which react a lot, the reactive oxygen. Oxidative stress may be caused by radiation, infection, pollution, aging, and also acute muscular effort. So a bit of physical activity is good, but don't overdo it, because this may also lead to, for example, with professionals, sports people, this does happen. We have a number of defenses against oxidative stress. We have enzymes which neutralize or counteract them. They're called also free radicals. And it is when these defenses are insufficient that at that point we suffer from oxidative stress, which may lead to modifications in terms of the lipids, fat, proteins, but above all, the DNA. And that's very dangerous because that may lead to mutations, and that will lead in turn to cancer, but also to neurodegenerative diseases with chromosomal breakages. Likewise, oxidative stress is going to generally weaken our immune system through a molecular mechanism, which I shall not describe now in detail. This, in turn, activates genes that are, on the whole, genes that code inflammatory molecules. So oxidative stress is caused by inflammation, but it then also causes inflammation. And this leads to an immune dysfunction of our immune system with apoptosis. As far as natural antioxidant defenses are concerned, there are a number of products that we eat, vitamins, but also products that we manufacture, the ones in red. For example, we have glutathione, which is a free radical scavenger, which we make. But at a certain age, we don't make it enough, and so we have to take supplements. These are naturally occurring non-enzymatic antioxidants. Then we have, clearly, there are also genes that are polymorphous, which means that some of us have very good enzymes while others don't. This explains why, for instance, there are some people who are smokers, tobacco smokers, and if they've got good enzymes, they can smoke, and they don't feel the negative impact of tobacco, in particular for cancer. But if we have bad enzymes, we can be hypersensitive, and even if we have passive smokers, we might develop cancer. So this can be analyzed in the lab, and we can have the information on the person. We can tell the person that they are at high risk or low risk if exposed to oxidating products. Here, I'm going to give you a few examples of antioxidants which one can use. They're not toxic. This is glutathione. It's a very small molecule, very active though, and also an extract of fermented papaya. I'm sure you've heard people talking about it, and this extract is biofermented and contains a number of products which are both immunostimulating and antioxidants. I'm going to give you two examples of the type of activity that they have of nutritional regulation of wound inflammation. Experiments were carried out on mice. The objective was to determine the effect of oral FPP supplementation. These mice have been modified to be obese and diabetic, and every day in water they take this product, which is an extract of fermented papaya. And we observe, we compare, for example, the impact that this has on the wounds, because we know that with the diabetes, wounds tend not to heal. This is true with human beings, but it's also true with mice. So very rapidly, I'm going to go over the details, but wound healing, as we know, is impaired in diabetic mice. And here you see the controls versus the diabetic mice. The diabetic mice, as you can see, don't heal as well unless they take papaya extract. And if they do FPP, they take FPP, well then their wound healing improves. Likewise, they can also control their fats better, their cholesterol, and even their glucose. Although papaya extract contains sugars, it does not increase the chance of developing diabetes. Another example, which is more recent, was carried out with people who are hypersensitive to electromagnetic fields, in particular mobiles. It is not a thermal effect, it's linked to the radiation itself. And the studies which were carried out in a large hospital in Paris show that these people have an oxidative stress. And it's the effect, as I said, of the electromagnetic radiation, which induces the oxidative stress with these people. And here you see there are various markers of the BSO, and it shows that following a treatment with papaya extract, it disappears after 3 months. And here you can see with imaging, it's called an encephaloscan or a brain scan. To the right, you have the restoration of the cephalorachidial liquid compared to the left of a patient who was hypersensitive to electromagnetic radiation and therefore had problems with that. This is just to give you 2 examples of the type of treatment which is absolutely non-toxic and which may, at least partially, restore the immune system and neutralize oxidative stress. This is a symptom. Clearly, this is not the cause, but it is a symptom which can be treated and improve the condition of patients who suffer from the stress. Now I'm going to move on and talk about the causes of these diseases. We realized that in many of these diseases, which were not known as infectious diseases, in fact, there are latent infections, especially bacterial latent infections. So here we have studied a new technology, well knowing, first of all, that bacteria are adapted to our treatment and took on forms that are resistant to the treatment itself. This new technology is based on the fact that some of the DNA has a bacterial or viral origin, can emit very low frequency electromagnetic waves. This is a resonance emission. The energy doesn't come from the outside, but from the inside, but from the outside. Through the environmental electromagnetic noise, we are surrounded by this electromagnetic noise, and here we see an effect which may be very damaging and which may thus increase the risk of chronic disease. So what we do is that we record in DNA solution, we record an electric signal which is amplified and then analyzed by computer to see its frequency and amplitude. How do we do this? By subsequent dilutions of DNAs, starting with a large dilution, 2 nanograms per milliliter, and by by a factor of 10, we dilute, and with the dilution, we observe a signal which has a higher frequency as compared to the excitation signal, which is from 7 to 100 hertz. And here we have a signal at certain dilutions which disappears at a higher dilution. Here you can see the example of a signal recording, which is background noise, and that has been recorded. Here we have discovered that only certain sequences, some sequences of viruses and bacteria, which are not very long, which is only up to about 500 base pairs, emit this signal after filtration, and that the structures that are in water haven't got the same, are not of the same size. Well, they vary according to whether they come from viruses or bacteria. So we can separate them by filtration, and the larger ones go through the filters of 100 nanometers, and we have 20 nanometer filters, while those that come from viruses, they also go through the filters of 20 nanometers. Likewise, to observe these signals, you have to shake each dilution very strongly, and also in a vortex, we have to have an excitation from the outside, which is something that we can generate or that some were found in the environment. We must also know that outside human activities, the Earth, our planet, has on the surface an electromagnetic, very low frequency network, which is of the Schumann frequency. The signals that we observe are between 1,000 and 3,000 hertz. Here is an example of the amplitude, which is a magnified amplitude of the signal in relation to time, but also here, through a Fourier transformation, you can analyze the various peaks. And here you have background noise, and you see the main is in orange, and we see whether it corresponds to electricity. And yes, there is a very weak electromagnetic field, but we are exposed throughout our lives to this field, and experiences by our Italian colleagues in Bologna showed that if the rats are exposed all their life to this radiation plus another radiation that induces cancer, the number of cancers doubles. So it is not totally innocuous or risk-free. And here is an example of a positive signal down below, where we have a much stronger wave between values of 1 and 3,000. We have been able to show that size is important according to and depends on the origin of the sequence, and biophysical studies carried out with a Japanese laboratory showed that in the dilutions of DNA that emit these signals, we have changes in the spectra of the water, especially in the near infrared. So these are really material structures that exist in water, and our colleagues, in particular Professor Del Giudice in Milan, a physicist, and Professor Vitello in Salerno, think that these structures are extremely stable and may last for hours and even days. And they have also shown us that they can store the genetic information of the DNA. That is to say, the sequence is respected and reproduced by the signal. So the structures in water that we call nanions, and also the electromagnetic signals which are emitted by these nanions, carry specific DNA information, and therefore we can record them on a computer, the waves, and also send them at a distance and reproduce the DNA sequence. So we think that there is a formation from the double helix of the DNA of a network of water molecules which are like a liquid crystal, and when they are diluted, they emit specific signals. So things are a little bit more complicated. I've simplified them in that in this emission there are waves that are shared by all DNAs, but as well as that there is a modulation of this wave according to the specific DNA sequence. What is interesting is the medical use of this, whatever the interpretation of all this. There's a lot of research that is required, but what is interesting is that we also find these signals of people who are infected. And we see the signals in the blood. We have pure cultures, different bacteria. You see the list of the various bacteria here, and all of them generate signals. Likewise, a number of viruses, especially the HIV-1, which is the AIDS virus, but also hepatitis B, and even the flu virus. In certain cases, we have identified the sequences, and in particular, in the case of the AIDS virus, which we will be coming back to in a moment. You might ask the question, what is the use of these signals? We think that they have a role in the induction of chronic diseases, and they make it possible to amplify part of the genetic information of the DNA, which may therefore lead to the formation of structures in water, billions and billions of structures, and that these, when they find a receptive cell, they reproduce the DNA, which again is going to carry new bad information back into the cell. You should also note that these structures are invisible to the immune system and therefore may not be affected by any of the chemical treatments. What is interesting is that not only people infected by bacteria, classical bacteria, for which there are already treatments, but also diseases which are not considered of infectious origin give signals in the blood of these people. This, for example, is the case of neurodegenerative diseases, Alzheimer's, Parkinson's, multiple sclerosis, various neuropathies, chronic Lyme syndrome, autism, rheumatoid arthritis, and types of cancer. So here we have a large field of investigation to find for each disease which are the bacteria that are involved and how they may come from the gut, for example. If you say bacteria, this implies antibiotic treatments, long-term treatments, and clinical tests. And some of our clinical colleagues have shown that you can improve the state of these diseases in certain cases through antibiotic treatments, but also through these others. Here, let me give you an example with Alzheimer's disease. Here we have signals in a signal of a patient. In the case of these diseases, one can imagine that there is a passage of bacteria from the gut because the mucosa is involved through inflammation and changes in the bacterial flora of the intestine, and this makes it possible to have a passage of the nanostructures in the blood. They may reach the brain, And lastly, they may also induce lesions at the level of the neurons. This is obviously a working hypothesis, but it makes it possible for us to think that in the future we may be able to prevent at the onset, before the final lesion or the final disease has taken off, with relatively simple treatments with antioxidants, antibiotics, and also changing nutrition. This gives us a hope to diminish the weight of these diseases in our society, that is to say, neurodegenerative diseases. Now I would like to conclude talking about a very special case, which is that of AIDS, HIV, and therefore talking about the HIV virus. In this case, we have discovered that the nucleic acid, the HIV RNA, does not generate any signals, while the form which is incorporated into the chromosome, that is to say the HIV DNA, does emit signals. What is interesting is that these signals appear especially with patients who've been treated. These patients have no more virus in the blood, but they have a sort of reserve of the virus in certain tissues where the DNA of the virus is hiding, so to speak. I would just like to remind you that this virus is extremely complicated in its genes. There are very many genes, and we would like to identify them. We have to identify them through PCR. The polymerase chain reaction, which means that we can amplify thousands of times a particular gene, and this is what we use normally. We use PCR for sequences for the end of the virus, which we called long terminal repeat, LTR. The current situation today is the following. Here you have a patient. We have HIV has become a chronic infection, and after a phase of multiplication, which peaks, the immune system is not yet immune depressed, and so it reacts, and therefore the amount of viruses or the amount of the increase decreases. But in time, this protection will not be strong enough, and the immune system will decline as the virus is going to multiply in the lymphocytes, which are the, so to speak, orchestra director of the immune system, the CD4. And at this point, the virus starts again to multiply. And as you know, very active inhibitors, which act on the reverse transcriptase. Here you have examples of the protease inhibitors. These are well known by patients. Well, the multiplication or duplication of the virus is virtually totally inhibited, except for the fact that, unfortunately, the virus is going to hide in certain tissues. We have not yet identified all of them or them, and the aim of our research project is to do so. That is to say, to identify where they hide and to try and eliminate them. What we have in green here is that this cache increases in time, so this reserve is virtually, we can't inhibit it by the virus inhibitors or using the virus inhibitors. We've tried to know why. Here, this is simply an image to show that if you stop the treatment, the reservoir remains in the middle, and if you stop or discontinue the treatment, it then regenerates the virus. So in summary, what we have discovered is that HIV DNA can be regularly detected in the plasma of patients treated by antiretroviral therapy and also in the red blood cells. detection can be obtained by several means, both with a PCR, nested PCR, but also with electromagnetic signals. This means that we have two methods that give the same results and which are biomarkers of the reservoirs of the virus. If we see that the electromagnetic signals disappear, and if we see that the PCR does not detect any DNA, we know that we are very close to a total eradication of the infection by virus. This is very important because the treatment is very expensive, and they must be taken every day by patients for the rest of their lives. And you can imagine that it is something very difficult for the patient to withstand, with secondary effects, very expensive for society, which explains why in many countries, especially developing countries, this treatment is not given to all patients. So the aim is to be able to see, when we identify the reservoir, to make it possible to have short treatments, 6-9 months, for example, as in the case of TB. And then after that, we could discontinue the treatment without the virus returning, leaving the virus at a very low level. We have already reached preliminary clinical trials to destroy the reservoirs. We have been using natural South African products which have antioxidants and immunostimulating effects. We think that maybe the immune system is going to help us to eliminate this virus altogether. So we compared patients treated with the ARV. Then we discontinued for a month to see if the other treatment was as affected and replace it. That's the first arm. And that is not so. That is to say that the natural products of a herbal origin haven't got the same effectiveness of the chemical antiviral, antiretroviral treatment. So the virus reappeared, and which is why we reverted after a month to the chemical-based treatment. Arm 2 was both, and R3 was control, so where they received ARVs, antiretroviral treatments, and the signals were still there, and the viral DNA was still there, both in the plasma and in the red blood cells. Num 2 is the most interesting. That is to say, we continued with the antiretroviral treatment, but we also added the extract herbal treatment. And in this case, we observed a decline of the signals and also in the PCR. This is very encouraging because we were able to show that we're acting partially on the virus, on the viral reservoir. One must also add, you can't see it here on this slide, that we have also identified a signal from a bacterial DNA. Apparently, in the same cells that have the viral reservoir, there is also a small bacterium. And this leads us to think that one could also treat this bacterial infection, for example, with antibiotics. And I wanted to show you two examples of signals which are different dilutions of the patient. Here you see in the viral DNA from plasma, these are months. 0 is where we started positive signals, and with the treatment as you can see, the signals become very, very weak and undetectable after 3 months. Here, you can observe the same pattern, but this is with bacterial DNA from plasma. The two seem to be associated, and I believe that the additional treatment that we're going to try now for a longer time is going to act both on the viral DNA and on the bacterial DNA. Here's an example of the PCR, but it's not very visible. Unfortunately, it's not dark enough for us to see it, but normally there is a band at 0 time which disappears after 3 months. In conclusion of these preliminary tests, we can say, which is on a very small number of patients, we can say that the herbal extract has no further inhibitory effect on HIV multiplication in patients already treated by ARVs. However, when added to patients maintained on ARVs, it decreases significantly the viral DNA reservoir in 5 out of 7 patients. And this is shown both by EMS decrease and PCR decrease. We find these results encouraging and will be continuing. And the hope is that if we manage to suppress the reservoir, one day we will be able to interrupt treatment and therefore without the virus resurging. This is new technologies, but we can also apply it to all the chronic diseases where we have these electromagnetic signals, and in particular with an antibiotic antibacterial treatment, which is also immunostimulating is immunostimulating in the long term for a long time. This is a great hope for HIV treatment, that is to say that the DNA will disappear and that the co-infecting agent is identified, and that therefore we will reach. It's a hope. It's not yet a fact. But we will eradicate the viral infection. I believe that more generally, with this technology, we can use it for blood transfusion safety, because if we have unidentified signals, we have to be careful. Obviously, for the identified viruses, there are tests, very good tests, that are very sensitive, but we could also diagnose this thanks to the non-identified agents, and this may cause long-term diseases. We might also identify hospital infections or nosocomial infections in the early stages. And therefore detect and fight all these chronic diseases. We don't necessarily want to prolong life. This is a possibility. We haven't yet reached our genetic outer limit, but the hope is to decrease the burden of these chronic diseases which handicap elderly people. It's a solution both individually, we should live a long time but in good health, but also for society, as this makes it possible to imagine a new model of social security, of public health, where you prevent all these diseases before they become irreversible, thanks to laboratory tests, and also, well, this also presumes that doctors have to learn a lot about these preventive medicines, and that we ourselves should have the approach to see, whereby we see the doctors before we're sick, and we should accept from a certain age to have tests, yearly tests or 6-monthly tests, to identify in the early stages these diseases. And also, I believe we should introduce new treatments using these biomarkers, not only in traditional medicine, not only for antibiotics, but possibly for electromagnetic treatment to neutralize these negative impacts. And the hope is also that HIV/AIDS should disappear in the course of the present century, and I hope I'll be able to see that. And with this, thank you. I would like to indicate that part of this work has been carried out, has been published. Here you see electromagnetic signals, electromagnetic detection has also been published, and that a lot of this research was carried out thanks to centers which are foundation established in Africa. This is the HIV/AIDS Ivory Coast Research Center in Abidjan, and I would also like to thank all those who have worked with me, all my collaborators who have taken part in this work. Thank you very much.